Yeah, I'm here.
Hi, Hassan. Great.
Hi.
So, Hassan, while I introduce you, why don't you get your screen share set up?
Hmm.
So, Hassan's a staff scientist in the Institute of Parasitology Biology Center, affiliated with Julius Lukas's group at the Czech Academy of Sciences and Faculty of Science, University of South Bohemia. So he will be telling us about crista diversity in eukaryotes, by which he must mean protists, and what they say about that very well-known protist humans and its mitochondria.
Exactly. Yeah.
All right, take it away, Hassan.
Yeah, thank you very much. Thank you very much, Andrew, for the opportunity to give the talk here. This talk is really about or centered around a review that I recently collaborated with Tomasz Panik, Marek Eliash, Maria Vanceva, and Julius about the diversity of Christie in eukaryotes and, of course, especially protists. And so also I wanted to use it as a platform to discuss some stuff that we had to leave out of the paper due to space constraints, and this is something that we're all working on, elaborating on right now. Okay, so I'm just going to start with, just so that we're all on the same page with sort of the basic idea, sort of going back to the meme that's been spreading around the internet, that mitochondria is the powerhouse of the cell. This is a meme that has emerged to show that schools are teaching students useless information, and I personally find this particular meme somewhat offensive because I do have some sort of affinity for the mitochondria, as I hope, I assume all of us in this group do. And right here is just a scheme showing the very basic, very basically how the respiratory chain works. So essentially, what happens is that complex carbohydrates are broken down by the citric acid cycle to produce reducing equivalents to feed electrons into the electron transport chain, complexes 1, 3, and 4. It's the flow of these high energy electrons through these different complexes that allows the pumping of protons from the matrix to the intermembrane space, which produces potential energy, which upon dissipation is used to synthesize ATP, the chemical energy unit of the cell. And this is all well and good. So this is a picture, this is a picture that was taken from Wikipedia. So this is a very, very generalized picture of what's going on. But as I'm sure many of you recognize, it's perhaps a little bit oversimplified, and it's missing one vital component of this whole system, which are the mitochondrial cristae. So these are the invaginations, or the infoldings of the inner membrane, that were observed in the 1950s by Palade and Suostrand. And basically, these are features that are found in aerobic mitochondria, and as is shown here, as is depicted here, the Christie membranes are enriched with the respiratory chain complexes. So here are the electron transport chain complexes, which are here, and complex 5 here. So so actually the Christie represent the powerhouses of the mitochondria. And furthermore, nice work done by Orion Shirihai group last year shows that Christie truly accumulate a lot of potential energy. So this is an image showing super resolution super resolution microscopy of mitochondrion from HeLa cells stained with a dye called TMRE that accumulates based on membrane potential. And what you can see here in this image, this is just basically taking the fluorescence data here and making a kind of a heat map. And the darker color shows regions of higher membrane potential. So clearly the Christie have much more membrane potential compared to the part of the mitochondria that is around the inner boundary membrane. So the Christie truly are the bioenergetic units of mitochondria. And so this is kind of encapsulated, or this is summarized here in this scheme showing the Christie membranes that are populated with the super complexes of the electron transport chain as well as ATP synthase. And so as you can see here, the Christie also encapsulates cytochrome C, which is the lone soluble component of the electron transport chain. So all in all, it serves to really encapsulate this whole system. And furthermore, one feature that helps to do this is Christie Junctions, which are shown right here. So these are the necks at the part of the Christie that are emerging from the inner boundary membrane as the Christie extend into the matrix. And these are small constricted spaces that serve as diffusion barriers between the Christie lumen, which is here, and the intermembrane space, which is here, as well as the Christie membrane and the inner boundary membrane, which is here. So in this way, the electron transport chain and all of the components of the bioenergetics of the mitochondrion are encapsulated in the Christie. Furthermore, so the Christie are not static parts of the cells. So what we see here is a depiction of respiratory measurements of isolated mitochondria. And what we see here is a line showing oxygen consumption. When ADP is added to the system in a molar excess, stimulates respiration. And this is called state 3 respiration. It's an active form of respiration. And when this ADP is exhausted and transformed into ATP, this oxygen consumption is leveling off and it becomes state 4 respiration. And in the 60s, Hackenbrock has actually took these different forms of mitochondria, or he took isolated mitochondria and treated them with ADP and looked at the morphology. And what he found was that in state 3, these actively respiring mitochondria have a condensed appearance, which corresponds to this very electron-dense matrix and wide Christie, whereas the state 4 assumes this orthodox appearance that we are all familiar with when we look at electron micrographs, where we see smaller Christie and slightly less electron-dense matrix. And what this study showed, even though this is a very artificial system, because you very rarely see or ever see condensed Christie in tissue samples. But what this did show is that Christie are very dynamic structures and their morphology does react to the physiological state of the organelle. And so this type of knowledge actually informed later studies showing that Christie remodeling is a vital part of things, processes such as apoptosis, which involves the release of cytochrome C into the cytosol, which is a potent effector of the apoptotic, one of the apoptotic pathways. When we look at mitochondria in mammalian tissues, we already see quite a diverse array of Christie shapes and forms, although they're mostly of the lamellar form. But of course, this is nothing compared to the diversity of Christie that we see when we look outside of mammalians and other metazoans and into other eukaryotes, such as Jacobids. Here's a picture of a mitochondrion from toxoplasma with vesicular Christie. Here is a kinetoplastid mitochondrion with the typical discoidal Christie in stacks. Here is a plant mitochondrion with a vesicular Christie, and one of my favorites when we were writing the review, the Mesozoan Chaos Carolinis, which upon starvation forms this really amazing matrix, which was observed in 1959, and this beautiful line drawing sort of rendering what's seen here under 10, which actually turned out to be correct upon later studies using 3D reconstruction. But what, there's a sort of an array of these different types of morphologies that we see throughout eukaryotes. And one of the goals that we wanted to do in the review was to kind of come up with a system so that we can rationally discuss these different Christie shapes. And so Thomas Panik actually undertook this endeavor, and he looked at 200 micrographs from 226 different species, and basically observed the different types of Christie that are in different organisms. So here, this is brown algae showing the irregular tubular Christie. Here is a Christie from a mitochondrion from ciliate, showing the typical tubular Christie. Here is a branched tubular Christie that is observed in amoebozoan. Here is a vesicular Christie that is observed in an alveolate. Here is a discoidal Christie that is observed in heterolobosian. And here is the lamellar Christie that typifies opisthecons, and this is an image taken from chick neuron. And so what Tom did was he basically came up with this system which encapsulates these different morphologies or these different shapes that I was speaking about, and he divided them, he further grouped them into two different let's say major categories, which are flat Christie and tubular vesicular Christie. And this is work that extends upon something done by Serovin in 1993. This was published in a Russian language journal. Using this as the base, we further refined this to make it a little bit more simple, and we also took into account the valence of the Christie Junctions, which I talked about before, which he couldn't take into account because Christie Junctions weren't discovered yet. So we feel that Christie Junctions are actually a vital part of this classification system, and I will talk a little bit more about this later. So what Thomas did next is he basically took all of the different variety of Christie morphotypes that were observed in these 226 species, and he mapped them onto the current tree, eukaryotic tree of life. And that is seen here. So in blue are the various forms of the tubular vesicular Christie, and here in red is the different forms of flat Christie. And not surprisingly, probably to anybody here, this reaffirms that Christie shape is not restricted to a certain supergroup, so therefore it doesn't represent a very good phylogenetic marker. However, certain groups do exhibit an enrichment of certain shapes. So the SAR supergroup predominantly has the tubular vesicular form, although actually there are also some notable exceptions. So in SAR, specifically in Rosaria, there is the Cenoradia, which are within this group, which actually exhibit flat Christies, either lamellar lamellar type, or unassigned type. So these kind of represent interesting exceptions that might warrant further investigation into molecular molecules that could possibly underlie these differences. And one final thing that we did observe in this type of analysis is that tubular vesicular Christie appeared to be the predominant form of the Christie shape throughout here, throughout eukaryotes, at least in the data set that we looked at. So in the review, what we tried to tackle next was to speculate on molecules that could be responsible for Christie diversity throughout eukaryotes. And so we decided to focus on molecules that are conserved throughout eukaryotes. So here are the four that I will discuss today. So cardiolipin, which is a lipid enriched in the inner membrane, as well as dimers of ATP synthase, which are found at the tips of the Christie here, promoting the positive curvature that's observed here. The Mikko Complex, which I have actually been studying with, in collaboration with the group that I have in Julius's laboratory, and these are at the Christie. These are responsible for maintaining Christie Junctions, as well as these two dynamin-like GTPases called MGM1 and OPA1, which are restricted to opisthecons. And so going into cardiolipin, so this is just a figure depicting this non-bilayer forming phospholipid. Unlike other phospholipids, this actually has 4 acyl side chains instead of the typical 2. And because of this configuration, it tends to form this conical structure that is depicted here. And within the membranes, this conical structure promotes negative curvature. So this is obviously something that could very well influence the shape of Christie. And so one of the features of the cardiolipin is that the acyl side chains actually are quite diverse, and they actually are variable in terms of the number of carbons that make up these side chains, as well as the level of saturation. So here are a couple of double bonds that are depicted here in trans, but if these were in cis, these would actually, as you can imagine, cause the acyl side chains to be further kinked, and it would actually promote, or I would imagine it would promote even more of a conical shape here. And this is data that was taken from Marcus Keller's group in Innsbruck. And basically what he did is lipidomic studies of Arabidopsis, slime mold, Saccharomyces, and mouse. And in this study, he showed that these different eukaryotes actually have different configurations of acyl side chains in terms of their carbon number, as well as the double bonds that are there. So we speculate that due to this could be a contributing factor to the diversity of Christie shapes. Another very beautiful example of how molecules can influence Christie shape is caused by the dimers of ATP synthase. So this is the final complex of the respiratory chain, which produces ATP. And this dimerization is actually unique to mitochondria. This hasn't been observed in bacteria nor in chloroplasts. And what we can see here, so this is work that was mostly done by Werner Kuhlbrand's group. And they looked at dimers from different organisms, such as green algae, plants, of course, opisthecons, and ciliates, as well as euglena. And you can see here already that the dimers do form different angles, which could presumably be a factor that would influence the different Christie shapes. In addition to that, they come in two different major configurations: this V shape, as well as this U shape, which is observed in ciliates. And furthermore, certain types are actually offset. So whereas these dimers are planar, so if you look at it from the top, you see that the F1 moieties form a straight line. Other forms actually are somewhat offset. And this actually is probably better illustrated by these images. So these are cryo-EM images of isolated Christie membranes taken from various organisms. And what you can see here is these dimers actually form rows at the edges of the Christie, where the positive curvature occurs. And what's quite striking here is, so here in Saccharomyces, we see the typical V-shaped dimers that are here, which form rather loose rows. And this is in contrast to the offset forms of the dimers that we see in Euglena. So these form much more tighter rows that are seen here. So these represent a dimer pair. Over here is another dimer pair, and this offset nature of the dimer allows the rows to be packed much more together. And this kind of beautifully shows how the scoidal shape can be formed by these type of dimers, which are offset. And in the ciliate paramecium, what we see here is U-shaped dimers, which form a very tight row, and the offset nature of each dimer actually causes a overall helical pattern in the row, which is mirrored by the tubular Christie that you observe in these organisms. So this is a really beautiful illustration of how complex 5 dimers can actually shape Christie. Okay, and then so this kind of brings us to the complex that's close to my heart, the Mikko complex, which is at Christie Junctions, which are parts of the Christie of intense of negative curvature. And so several of the subunits of the Mikko complex have been implicated in doing this, namely a subunit called MIC10, which is conserved throughout eukaryotes. That's stuff that Sergio, along with Jeremy Weidman and others, have shown in their really nice study survey throughout eukaryotes. And so it's speculated that the oligomerization of MIC10, which forms this sort of wedge shape here with two transmembrane domains and a small loop of amino acids that are extending into the matrix. When these oligomerize, these wedge-shaped proteins form this negative curvature here. In addition to that, other groups have shown that the mitophilin domain of MIC60, another very, very conserved protein or component of the Mikko complex. In fact, this is the component that was this mitophilin domain has also been demonstrated to be able to remodel membranes in vitro. So presumably the action of both of these would help to form the negative curvature there. We actually studied the complex in trypanosomes. So this is the only Mikko complex outside of opisthecons that has been fully characterized in terms of all of its subunits. And there are some interesting differences between the well-studied opisthecon complex and ours. So one of them is the configuration of the conserved MIC10 subunit. So in trypanosomes and actually most other eukaryotes, this loop that I was talking about, which is in the matrix, is actually comprised of 6 amino acids, whereas in opisthecons, it is only 3 amino acids all of them positively charged. So this is one sort of difference between the two, between what opisthecons have and what other eukaryotes have. Furthermore, in the trypanosome complex, we actually couldn't find a protein that is embedded in the inner membrane with a predicted transmembrane domain that has the signature mitophilin domain, which I mentioned before. So we speculate that one of these other subunits that are soluble could represent this mitophilin domain. One other big difference is how the complexes are divided between opisthecons and trypanosomes. So in opisthecons, the Mikko's complex is actually divided into two subcomplexes, which are organized around the core MIC60 subunit, as well as the core MIC10 subunit. In trypanosomes, it appears that the subcomplexes are organized not laterally in the inner membrane, but into a membrane-embedded complex and a soluble complex that's in the membrane. And one more surprising discovery that we found in trypanosomes is that one of the subunits is a thyrodoxin-like protein, which appears to have a role in inner membrane or intermembrane space import. So this is kind of depicted here. And this also kind of shows our current model of how the trypanosome Mikko's complex works. So this thyrodoxin-like protein, we speculate, is responsible for the oxidative folding of small proteins that are imported into the intermembrane space and require the oxidation of their twin thiol groups for their oxidative folding and maturation. And many of these intermembrane space proteins are actually assembly factors for the respiratory chain complex. And so, so this actually fits nicely to the idea of how Mikko's can populate the Christie membrane with the respiratory chain complexes. Okay, and then finally, and sort of kind of getting back to the title of the talk, I'm going to talk a little bit about the dynamin-like GTPases, MGM1 and OPA1. I'm sorry, I'm going to go back into metazoans. So OPA1 is a dynamin-like GTPase that is responsible for remodeling of Christie in mammals. And so what is interesting about this protein in particular is that it actually has two forms, the long membrane embedded form, as well as a short form, which is produced by proteolytic cleavage. And so this is actually depicted here in this western blot here of OPA1, showing the long forms, which are big up here, as well as the shorter forms down here. And so it's the oligomerization of these two forms that is believed to actually create the tightness of the Christie. And when these OPA oligomers are lost or somehow depleted or reduced, the Christie become wider and eventually open up. And in terms of apoptosis, this releases cytochrome C, which I mentioned before is a effector of apoptosis when it's released into the cytoplasm, as well as other apoptotic factors that are trapped here. And in our study, we decided to just look and see whether the opening of these Christie, Christie junctions is significant. And what the graph shows is that upon action of apoptosis, the Christie junctions indeed do open to a diameter that's actually greater than the diameters of Christie junctions that are observed in wild-type cells. And here, this 40 nanometer diameter is actually from Paramecium, again, the ciliate. So this is an unconstricted Christie junction here. So clearly, these openings are very significant. Okay, and then MGM1 is another dynamin-like GTPase that is found in fungi and related organisms. There was a very nice study done by Oliver Dumke's group showing how this actually works. So what they were able to do is, actually before that, I should mention that also like OPA1, it forms a long form, which is membrane embedded, as well as a short form, which is soluble, also produced by by proteolytic cleavage. And what we see here, and what this group is, they took advantage of the soluble short form, and they showed that in vitro and on membranes, they were able to form this crystal-like or this lattice-like structure. And by the GTPase activity, it was able to expand or contract the membranes, thus showing how these GTPases can remodel the membranes, the Christie membranes. And so in this paper and many other papers, people actually use MGM1 and OPA1 interchangeably because it's assumed that since these are from opisthecons and thus somewhat closely related eukaryotes, they must be true orthologs. And actually work by, once again, in the paper that Sergio and coworkers published in Current Biology, they actually shed some doubt into the monophyly of OPA1 and MGM1. And so with Marek and Tomish, we decided to look into this a little bit deeper. So in the literature, there is some evidence that these might not be true orthologs. So for one, if you look at the primary structure of the two proteins, what's immediately apparent, especially to Marek, who's an expert on these GTPases, is that the GED domain, the GTPase effector domain, is highly divergent in OPA1 compared to MGM1, which is already very strange. Furthermore, these two proteins, even though they have a very uncanny similarity, appear to be processed, or they are processed by two different proteases. MGM by rhomboid, and OPA1 actually has two cleavage sites, and they are cleaved by either OMA1 or YME1, which forms a, which algorithmizes to form the intermembrane space AAA protease. And so, and actually, interestingly enough, when OPA1 is exogenously expressed in yeast, it's not, it is not processed by rhomboid protease, which further illustrates that these two proteins have very different properties indeed. And finally, it's very notable that the Christie junctions of yeast and mammals are very different. So mammals, mammalian Christie junctions have the typical tubular form that's found in most other eukaryotes, whereas yeast predominantly have these slot-like Christie junctions. So this may be due to the fact that these two proteins have a different mode of action. So what Marek and Tomish did is they actually did a phylogenetic analysis to really investigate the monophily of MGM1 and OPA1. And many of you know Marek is extremely thorough, so he actually went through many, many different gene databases, metagenome databases, and so on. And what they found was that indeed, when you align these two things, they do not form monophyletic clade, so they do not appear to be true orthologs. Interestingly enough, the holom Mikko appear to have MGM1, and OPA1 is actually found here in holozoan opisthecons as well as in the Crohn's group. And because Marek was extremely thorough, he serendipitously came across a third class of dynamins, which he decided to dub mid-X. And interestingly enough, even though a couple of sequences were found in sterninopiles or amoebozoans, most of the mid-X sequences were retrieved from the genome scaffolds of these megaviruses, these rather large viruses. So this is actually a very unexpected discovery, and right now we're trying to further investigate whether the prediction that mid-X, which are found on these viruses, is targeted to the mitochondrion, because as you see here in this scheme, it does share many features like MGM1 in terms of its GTPase domain, and many of these sequences also bear very robust predicted mitochondrial targeting sequences. Okay, so this discussion about MGM1 and OPA1, which are two GTPases that are restricted to opisthecons, coupled with the fact that we were looking at Christie from 226 species, kind of brought to our attention that that opisthecons actually might be the outlier here in terms of Christie shape and dynamics. For one thing, the lamellar Christie that are observed in opisthecons, when they were better resolved by electron tomography in the 1990s, it became apparent that the lamellar Christie were actually joined to the inner membrane by multiple Christie junctions, which are depicted here. So we, in the Christie classification system, we suggest that the Christie junction valence of lamellar Christie should be 1 or greater. And this is observed here by the pioneering work of Perkins and Ellismint and other co-workers in 1997, which first described these Christie junctions, and later on by Karen Davids from Werner Kuhlbrand's group, which showed these very nice tubular Christie junctions that I've shown already before. And in both of these papers, they were describing these lamellar Christie as sort of fusions of tubular Christie, which is a rather interesting observation indeed. And here in this corner, we see one of our own tomograms of discoidal Christie from trypanosomes, and there is only one Christie junction here. So it really is very consistent with this idea that the lamellar Christie are unusual in that they are really just these fusions of tubular Christie, which haven't been observed in other eukaryotes yet. Furthermore, when I'm sorry, we'll go back. Furthermore, when the lamellar Christie of the Christie of Saccharomyces was observed by electron tomography, the researchers observed that there were predominantly lamellar Christie, although they also found the occurrence of tubular Christie as well, which is consistent with this idea. When they basically used yeast, which actually have a temperature sensitive mutant, so of MGM1, they were able to ablate MGM1 function for a short period of time by raising the temperature, and they observed in these yeast the disappearance of lamellar Christie and the emergence of tubular Christie, which is somewhat consistent with this idea that lamellar Christie are really just fusions of tubular Christie. And this year, Andreas Richard's group, using super resolution STED microscopy, looked at HeLa cells again with these lamellar Christie, and over a second time scale, observed both fusion and fission of Christie. So it seems, although it wasn't clear whether these were tubular Christie that were fusing into lamellar Christie, or it is very much consistent with this idea that maybe lamellar cristae are really unique. Okay, so this kind of brings me to my conclusions. So cristae are the bioenergetic units within mitochondria. Their shape often reflects the physiological state of the cells. Cristae are observed to assume different shapes throughout eukaryotes, and Thomas Panik classified them into two major categories: a flat cristae or tubular vesicular cristae. And so it kind of begs the question whether these shapes can somehow affect the bioenergetic state, or whether this is a reflection of the bioenergetic state of these cells, or maybe a little bit of both. And then what we also did was speculate on some molecules that could underline these different shapes. In particular, the ATP synthases look like very good candidates for doing this. And finally, and this is somewhat unusual and refreshing for me, to say that opisthecons might be the special case among eukaryotes in bearing these lamellar cristae that might represent fusions of tubular cristae. And if so, and also these GTPase, this dynamine-like GTPases, OPA1 and MGM1, which do not appear to be true orthologs of each other, whether they actually do something specific for their respective lamellar cristae in humans or yeast. I think that in the review, when we were going through this stuff, one thing that we really need is to do some electron tomography on the mitochondria of other eukaryotes. So opisthecons are vastly overrepresented in such studies compared to the rest of eukaryotes. And in fact, we added a couple of points to that in our Mikko study. So I hope that this also encourages people to apply electron tomography to their favorite protists to reveal what type of shapes that they really have, and put this model that we have to the test. And if you're interested in more, please look at our, please look at our paper that should be coming out sometime soon in print in Current Biology. And okay. Okay, for some reason I am stuck. Okay, and with that, I would like to conclude by acknowledging my co-authors in this study, who I really enjoyed collaborating with: Thomas Panik, Marek Eliash, who never ceases to amaze me with his knowledge; Maria Vanceva, who collaborated with us on the electron microscopy; and of course, Julius, who also I like to thank for encouraging me to take this free slot. I also want to acknowledge the members of my team: Joseph Karoff, Rudy Cadena, and Ivy Heller, who all were co-authors on the Current Biology paper about Mikos, as well as Nina Sheikh, who is a new member of the group, and we expect great things from her. And the Mikos study was done in collaboration with Andre Schneider, which was also a very wonderful collaboration. And finally, since I'm sharing this session with Sergio, I'd also like to thank him. Last summer, he had a research stay in our laboratory, and it was really great having an opportunity to talk to him face to face. And with that, I think, and also I'd like to thank our funding agencies, and thank you for your attention, and thank you once again for giving me the opportunity to give this talk.
Okay, thank you very much, Hassan. That was very interesting. We're a little bit over time, but I think people who have hold on still want to ask questions. So please take away the questions. If anyone has any thoughts, please share them. Questions? Okay, I'm going to ask a question, which is this OPA1 versus MGM1. Which one did you decide, in that paper, did you decide which one was ancestral to all eukaryotes? Because it looked like one was derived, MGM1 looks like it's a fungal thing. Is that basically?
Yes, so
okay.
exactly. So due to the fact that OPA1 is found in the crumb supergroup as well, I think right now we're currently speculating that MGM1 most likely replaced OPA1 in these fungal groups here. So it seems that OPA1 may have a deeper ancestry compared to MGM1. And it's kind of tempting to also speculate that maybe MGM1 was acquired in this fungal group from one of these megaviruses, but this is pure speculation.
So for most of those other eukaryotes on the Diaphoreticus side, we just don't know what's doing that kind of thing, or that you don't think they're doing that?
Well, there haven't been any GTPases identified from that group that were shown to be mitochondrial or have a role in Christie membrane remodeling. So it is still an open question whether these other groups have it. So one thing I kind of skipped over is Discoba. So if I go back here, so another one of these interesting exceptions that Thomas observed was that in euglenozoans, the discoidal Christie is a predominant form, whereas in the sister diplonamid groups, there appears to be what looks like lamellar Christie. So it would be very interesting to look into the diplonamids and somehow see whether there's GTPase like, or a dynamin like GTPase, or some other type of protein that could be responsible for potentially fusing discoidal Christie together to form these lamellar forms.
Thank you. So, floor is open for some questions. Well, I don't want to push people, but please, if you do have questions, please ask. I'm sure that Hassan would entertain questions if you email them to him as well. I know
for example.
It's getting very late there in Europe, so, oh, hold on, Jana Eaglet has a question. So.
Hey, that was a really cool talk. Thank you.
Okay, thank you.
Just out of curiosity, like you were encouraging us to go out and explore the sampling of mitochondrial Christie structure throughout. What goes into something like that? Like this is presumably through cryo-EM.
Exactly. Well, it doesn't. I think tomography would be sufficient for doing a lot of these type of studies. So the early work on the work that identified Christie Junctions in the first place was done using tomography. So obviously, cryo-EM would be a very nice and robust tool for doing that. But I think tomography would be sufficient for this type of work.
And you don't need to isolate mitochondria or anything like that?
You don't necessarily need to isolate mitochondria. So the tomography has been done on isolated mitochondria, but they've also been done on in situ. And in the paper, we actually compared the Christie junction diameters of isolated mitochondria versus mitochondria observed in situ. And there were slight differences, but they weren't statistically significant. So it doesn't appear that observing them one way or the other would really affect the overall morphology of the Christie.
Interesting. Thank you.
Thank you.
Okay. Well, I think it's come to that time of the evening that we can decide to say Thank you to our speakers. Thank you, Hassan, for a real tour de force through mitochondrial Christie, and it makes me want to go and have a look at the paper in detail and digest all of that incredible information that I had no idea existed. So thank you very much for that.
Thank you.
So let's just give a round of applause. 3, 2, 1. It's hard to coordinate these things. And what I will do is just say that we'll see you in a couple of weeks after the 2-week coffee break, and where we will be able to see Daniel Richter give us a nice talk on microbial eukaryote biogeography and gene expression in the open ocean. So we'll move towards a microbial ecology change in the topics. So I look forward to that, and thank you both Hassan and Sergio for great talks today, and I hope everyone's well, and enjoy your sleep, those of you in Europe, and enjoy your day, those of you in Japan, and I'm going to go enjoy a beer.